Okay, should have heard a little notification saying this session is being recorded. Uh, all of our virtual taster lectures are being recorded and they will be shared afterwards, but it may take a little while to get them uploaded. But yep, so it's 12:30. I can see people logging in, so it might take a few minutes to get everyone on board. So whilst we're doing that, I'm just going to do a few things to get us going. First of all, if you can, on whatever device you're on, if you're on a desktop or a mobile, you should have a little row of buttons which allows you to change the configuration so you can see speakers in a different format or slides. But you've also got a chat function, a Q&A function. Could you, if you can hear me, just pop in the Q&A, pop in the chat function and say hello, hi, just let me know that you can hear okay. That'd be great. Great, thank you very much. Fantastic. It's always really good to know that the technology is working and that you can hear us. That's great. Good. Fantastic. So the numbers are slowly climbing. So just a quick. Oh yes, somebody said hello from Germany. Fantastic. Could you actually let us know if you're happy to? Let us know where in the world you're joining from. So far in this series of taster lectures, we've had sessions kind of earlier in the day and later in the day, and this is a lunchtime lecture, so it's interesting for us to know where you are, where you're joining from. It just gives us a feel for what sort of time zone is good for what kind of attendees. And I can see here we've got people from Germany, a couple of people from London, some from Romania, and some from India, and some from Denmark. So very interesting. And Nottingham as well, and Ithaca. So that's fantastic. So somebody is getting listen to this whilst they're getting ready for work. Fantastic, and somebody from Spain. Well, it's great to have you with us. Thank you very much for joining us. It's fantastic to have you here. I'm going to go through a few little housekeeping things before I invite Ben to start his talk. But first of all, this session is being recorded, as I said already. You'll be sent a link afterwards at some point to watch this back and all the rest of the taster lectures that we've done. My name is Kim Morgan. I'm the Outreach and Communications Coordinator here at UCL Biochemical Engineering. Really important point to make is that we may, in these taster lectures, sometimes we go do a bit of a deep dive into some of the science or engineering. Someone from Curitiba. I'm very familiar with Curitiba. I have family in Maringa. So, yes, sorry, not relevant to the session, but anyway. Yes, so if we do go into some science or engineering, and some people have been concerned that they don't know that. If you come and do a degree with us, we will take you to that level. What we do with our degree programs is we will do that. So, what I'd like to do to start off with is to launch a poll and just ask you. If you're considering studying, what sort of study that would be. So would it be that you're more interested in doing a degree like a BEng or an MEng or a postgraduate? It really helps us to gauge the level of our audience, and it's very interesting for this one, because in previous sessions we've had like 50-50 or more prospective undergraduates, more prospective postgraduates, and it's really interesting that for this session here we've got slightly more prospective people who'd be interested in doing a postgraduate degree, so an MSc, which is very interesting. It's a slightly different balance from some of the other sessions. So thank you very much for that. And the other question that I'm going to ask you now is, I'd just like to know if you have an offer to study. It's because one of the things that we do is we get in touch with people who have an offer for a degree, and we ask them whether they'd like to go and join these, so they can find out more about our department, find out about our research and some things we're involved with. And I can see that the majority of people who are applying don't have an offer, but we do have some, which is very helpful. Thank you very much for doing that. Okay, so somebody else has got an offer, but not at UCL. Very, very interesting, very good to know, and it's great that you've chosen to join us. Fantastic, thank you. So, thank you for filling those in. Just a few things about how this session is going to work. First of all, you've already discovered the chat function, I hope. Some of you let me know where you're joining from. If you have any technical problems, if you can't hear us clearly or you can't see the slides, please do let us know in the chat. Myself and Ben will be able to see that. And if there's any kind of technical issue, you can let us know there. We're really looking forward to your questions. Please ask anything you want to related to the subject or what it's like at UCL Biochemical Engineering. That's fine. But you can please use the Q&A function. It's really helpful for us. Using the Q&A function means that we can see the question. You can ask them at any point in the session and we can see them and they can build up. It's easier for us to answer them, and it's a lot easier in the chat because sometimes the questions can get lost in the chat, whereas the Q&A, it's a lot easier for us to queue them. We can mark them as answered and things. So please put your questions in the Q&A box at any point in the session, but we will answer them coming towards the end. So I think that's all the housekeeping out of the way. Ben, if you're ready, would you like to turn your camera and microphone on? Fantastic. Okay, this is my colleague, Dr. Ben Dages, and I'm going to hand over to you for this. I might turn my camera off temporarily, but I am here in the background if there are any technical or anything that you want to know. Otherwise, Ben, it's over to you.
Cool. I need to share my screen.
Sure. Let me just stop sharing. There we go. All yours.
Okay. So, hello everyone. My name is Dr. Ben Dages, and today I'll be asking how soon before lab-grown meat becomes just meat. So first I'm going to be giving you a little bit about my background and then introduce cultivated meat in relation to alternative protein. Then go through all the steps that have brought us to where we are today, and then try and look at the current challenges that we have and the bottlenecks and how we're trying to solve them at UCL. Finally, I'll be talking a little bit about how you can get involved if you do end up joining us here at UCL. So as I said, I'm Ben Dages. I'm a cultivated meat specialist. I did my PhD in cultivated meat, which is quite rare. And I'm now doing a postdoc also in the same field here in the biochemical engineering department. My specialty is making scaffolds, which are supports for cells that can grow in 3 dimensions and that can be integrated in the cultivated meat by a process to make cell expansion better or cell differentiation more efficient. And more importantly, they are edible, so they can be part of the final product. So why do we need alternative protein and cultivated meat? Well, the thing is, there's a bit of an issue at the moment. The global population is on the rise. And it's even being expected to reach almost 10 billion by 2050. And with that population increase, the demand for meat is also on the rise. It's actually almost exponential. And it's been expected to double from its 2000s levels in 2050. So how can we supply such a high demand for meat? Well, farmers can only really operate as factories in a process called intensive farming. And really what it is, it's a high input, high output type of system, which is extremely damaging on the environment, which is damaging, which puts a strain on food security and public health, and also is obviously terrible for animal welfare. So it's really important that we get sustainable protein alternatives. So there's different classes of protein alternatives that are more or less available today. First, there's the plant-based one. So plant-based are basically Beyond Meat, any kind of veggie burger. They are basically plant proteins that have been crushed and made into a meat product. These are quite popular. They're available everywhere. And recently, work has gone in to try and engineer the sensory properties of them so that, it looks like meat, it feels like meat, it tastes like meat. Then we also have the fermented ones. These are mainly mycoproteins. So you're not necessarily eating mushrooms, but you're eating the protein that mushrooms produce in a bioreactor. These are also quite popular. Like corn would be an example of these. And this relies on a field of study called cellular agriculture. Then we have insect protein. So insect protein, according to some people, that's not really alternative protein because you're slaughtering insects, but it's another way of getting animal protein in a very environmentally friendly way. Generally, this would be a hybrid product, which would be there for insects and plants, or insects and mycoprotein, but not necessarily. The only big problem with insects is that, at least in the West, no one wants to eat them, and it really evokes disgust. And then finally, we've got cultivated meat or lab-grown meat. Cultivated meat is the newest of all of these, and it's animal protein without animals. And so instead of farming animals, we just use a stem cell that we will grow and differentiate into the components of meat, all in a lab, all safe, yeah, and it will be cellularly and taste and nutritionally exactly the same as real meat. But how do we get here? Well, there are many steps that have brought us to where we are today, but what really helped was research that was done about 100 years ago on the first tissue culture experiments. So it all starts in 1885, and there's a guy called Willem Roux, and he manages to sustain chicken cells alive for a few days in a saline solution. It's not growing, but it's not dying. But in more or less, and 10 years later, there's a guy who it's the first time anyone's thought of this. And he thinks it might be possible in 100 years to be getting food made in factories. He didn't really explain how he would do it, but it would be technically an artificial type of meat. Anyway, tissue culture well, cell culture is expanding. And by 1907, Harrison invents the drop culture technique, where you can basically have a culture upside down and create and expand the cell. So it's the first time we actually gain some cell expansion. But this doesn't last very long, and you can't really make any tissue from that until a guy called Alexis Carrel comes in, and he manages to immortalize cells. Now, when you have immortalized cells, they can grow forever. You can just passage them over and over again. And this culture went on for about 30 years, way longer than the life of the chicken that it came from. And actually, in 1922, one of Carrel's colleagues said that if all of the cells had continued to expand for that amount of time, all of the passages, all of the cells, well, the weight of these cells would exceed the mass of the sun. So quite a lot. So then throughout the 20th century, there's a lot of scientific advances that enhance tissue culture or make it actually possible to grow tissues outside of the lab, outside of the body, sorry. And also a few wild predictions that start to shape how cultivated meat might look like in the future. So the first few things were the possibility to actually grow tissues in sterile flasks. That happened in the 1930s. Then in the 40s, we discover that actually, if you grow tissue, well, if you grow cells in 2-dimension, you're not really going to get the tissues that the way they look like in the body. So these have to be done in aggregates and maybe under a constant agitation. Then we discover stem cells, and stem cells, they are able to differentiate into different lineages. So a stem cell could become muscle, then fat, or bone, or whatever we decide. And meanwhile, while this is happening, one of the first people to have a prediction about what cultivated meat will likely be like is Winston Churchill. And he thinks that in 50 years from the 1930s, we'll be able, with the technology that we have in tissue culture, and with a little bit more knowledge in growth hormones, to actually be able to create meat in a lab within a small amount of time. We also have evidence from, not evidence, but we also have examples from science fiction. We have one of the very first depiction of cultivated meat from Ashes and Ashes, which is a book that happens in the future. And also in the 50s, we have a guy called Willem van Elen, and he survives. He during the war he survives the Japanese POW camps, and in those camps it's brutal conditions, brutal for the prisoners, but he also witnesses absolute brutality on livestock. And although he's not a vegetarian, he thinks that it would be really good if there was a way to continue eating meat without hurting animals. And so he'll dedicate his life towards finding a possibility to this. But he's really interested in what's happening in the engineering space and the tissue culture space, and he's inspired by the possibilities of of having these cells that become anything we want. And as we have development in bioreactors and in meat serum-free media, he realizes, well, now it's a possibility. It's not in the future, it can be now. And in 1999, he actually writes the first patent for cultivated meat, which he calls in vitro meat. So it's only really from 1999 that we start getting proper cultivated meat that we can smell or taste. So the first experiments are done by NASA. So NASA, for some reason, thought goldfish would be appealing for astronauts. So they grew these goldfish steaks, and then they say they smelt them, and they smelt like fish. From that, they said it was acceptable food, but they didn't taste it. Then a year later, there was a exposition where participants were invited to eat cultivated frog meat with the actual frogs living in a little terrarium next to the restaurant, next to the table. So you could see the meat that you were eating, see the animal from which you're eating the meat. We then have New Harvest. New Harvest is like a lobbying body based in America, but they are one of the first to really fund cultivated meat, mostly through academia, but also later on through startups. And then nothing until in 2011, a guy called Mark Post, a professor in Holland, has an interview, and he says, well, we actually have all of the technology to make cultivated meat now, but we don't have the investment. And with a little bit of help, well, quite a lot of help from Sergey Brin, who's one of the directors of Google, he manages just 2 years later to produce the first cultivated beef burger. And this was made at lab scale, so you can see all the culture flasks, and there are literally thousands of them. It was sold in London in 2013 at auction, but for the cost that it was made, for 250,000 euros. So quite expensive. And then from this, we get the age of the startups. We get, in 2016, 3 startups are founded. GFI is also founded, another body, a bit like New Harvest, which is going to help fund these startups and fund academic research. And from here, it's going to go really, really quickly. Memphis Meat, that same year, manages to create a meatball, which it claims only costs about $1,200 to produce, so 250 times less than the burger 3 years prior. And then 2 years later, we get the first cultivated steak. Although it's just 1 millimeter thick, it's only $50. And just 3 years from there, we get the first bioprinted proper steak that we can see in the picture. And that is also the same $50, but you get way more. We also now have cultivated meat grown in space. We also have Singapore, the first country to approve cultivated meat. At the time, it was just a chicken nugget, but it has since approved more products. The UK, the first country in Europe to approve cultivated meat, although it was just pet food. But here's my dog Truffle eating them. Finally, the newest stuff is that Australia and New Zealand have also approved Val's quail cultivated meat. So basically, getting there was just a mixture, interdisciplinary research of regenerative medicine, so creating tissues in a lab, cellular agriculture, making food from cells, and food science and technology, so optimizing the nutritional profile, the taste, and the texture. So I'm just going to go on and explain a little bit about how we make cultivated meat, or how people would make cultivated meat in a quite simplified way, and then talk a little bit about the challenges that we can face. So first, you need an animal. Here it's a cow, and you want to take some stem cells, ideally stem cells. So there are different types. You could get mesenchymal stem cells. You could get induced pluripotent stem cells. You could get precursor cells that can only become one type of lineage, or you could even just go from the embryo and get embryonic stem cells. You would then try and bank the cells or immortalize them if needed, and then you never ever need to go back to the animal. You then need to expand those cells, try and get as many as you possibly can, and then you need to differentiate them into muscle and fat or whatever components you want. And then there's two ways of going there to the end product. Either you're making minced products, such as burgers and sausages, and then you can just continue that differentiation just by changing the media, changing basically the food you're giving to your cells. Or you can remove your cells and then add them to a scaffold that will provide texture to the meat. And through there, you can make more complex structures that would make structured meat like steak or ribs or anything that would be a whole cut. Now, there are major bottlenecks that impede release to market. So cultivated meat, there's about 12 products out there, and yet it's impossible to have them. And one of the main reasons is that they're just not that scalable, and they're much too expensive for anyone to buy. Really, they need to reach cost parity for people to start considering changing their diets. Problem with the cost, it's mostly coming from the media that we use to feed the cells. But literally, there are challenges at absolutely every point in this diagram. From the cells, from the expansion, the differentiation, the scaffolds, the assembly, and all that. But I'm now just going to talk about the upstream, so everything to do with the cells. So in vivo, so in the body, cells live in their own microenvironment. They are surrounded by an interstitial fluid, which is a basically salty, sugary solution. They're fed through blood plasma, which has the growth factors and binding proteins, hormones for growth and differentiation. And also, these cells are generally considered adherent, so they need a surface to grow on. And that is called the extracellular matrix, which also provides some mechanical and topographical cues that would help differentiation. In vitro, when we're in the lab, we need to try and replicate these conditions. So we use basal medium to replace the interstitial fluid, which is mixed with serum, which comes from animal blood. And then we use scaffolds, or these artificial supports, to replicate the extracellular matrix. But the problem with cultivated meat is that we really want to try and get animals out away from the whole process. And so we need to find a way to replace the blood serum by something else. And that something else needs to be as simple as possible to help with the regulatory approvals. It needs to be really, really cost effective. And it absolutely has to be animal free. So now I'm going to talk a little about how you could make serum-free media. So the problem with the serum is that it's the most expensive part. So if there's a way to just change it, that would be great. But serum is composed of hundreds of proteins, mostly unknown ingredients, and it's going to be very, very hard to change that. We don't even really know what is important for the cell culture that is in the media. Some companies have made commercially available serum-free media, but we found that actually they don't always work on all types of cells, and also they may not be stable through a lot of passages. So as the culture continues, it might not be as efficient as others. So Will, who's a PhD student at UCL, has been trying to solve just that problem. So what he did, he tried to grow some cells using these commercial B8 and B9 media, and found that it didn't work too well. And so he then tried to analyze all of the ingredients that these medias have and just try every single condition separately to then find what worked and what didn't. He then got some food grade basal media and just added the ingredients that did work and found some other external ones that could maybe boost cell growth. He then tested it in static conditions and dynamic conditions with spinner flasks and actually found that his formulation worked really, really well. And so what his goal was is to remove the animal entirely. He used recombinant proteins, so proteins grown instead of from an animal, from bacteria or plants, and also some food grade inputs such as molecular crowders like spirulina protein, methylcellulose, or beta carotene. So then that brings us on to the scaffolds, the thing that replaces the extracellular matrix. So the extracellular matrix is really a nanofibrous, microporous mesh that surrounds the cells and keeps them together. You can see a picture here of what it looks like. And there are various ways that we can do this in the lab, but one of the most popular ways is through a technique called electrospinning, which uses a polymer solution connected to a high voltage, and it draws the polymer into a droplet and then into a fiber that will whip across the surface. And you can see a picture here of on the side of the whipped fibers, and they are nano in scale. And below that, we can see some cells that are growing on that surface. To choose your right biomaterial here, you really need to consider a few things. Firstly, it needs to be sustainable. Otherwise, it's going to be difficult to get more and efficiently. It needs to be edible because it might be part of the final product. It needs to be compatible with the scaffold design. So if you're going to use electrospinning, it needs to be electrospinnable. It also needs to be compatible with the cells, i.e. non-toxic, but also potentially help with the differentiation or expansion. And also they need to be stable in an aqueous environment because they're going to be in a bioreactor for a while and they must not dissolve in that time. And even if you get all the science right, they still need to be accepted by the consumer and the regulators. So these scaffolds can be used for two things. They can be used for expansion and for differentiation. And for the expansion, generally they'll be used as microcarriers. So they're these tiny little supports, little scaffoldings that you'll grow your cells on in small flasks, medium-sized bioreactors, and you have to go bigger and bigger through a seed train to be able to then expand them in gigantic building-sized bioreactors. These are generally spherical, and there are different materials that you can have. In tissue engineering, a lot of people use plastic, but otherwise you could use something that's food grade and covered with extracellular matrix proteins. Those proteins are generally quite expensive, and for cultivated meat, we try to do without. So this brings me to what I did for my, not for my postdoc, but for my PhD project. So I used silk fibrin, which is a protein from silk cocoons. And then I mixed it with zein, which is a protein from corn, and some psyllium husk, and then I electrospun it using the technique that I showed before, broke down the mat into very, very small pieces, and used these pieces as my microcarriers. So my microcarriers were not spherical, but they had this nanofibrous, microporous network around them, which helps in the cell attachment. I then compared my cells on these microcarriers with commercial microcarriers and found that mine were just as good despite not having any functionalization on top of them, so no extracellular matrix proteins. And also, when I was mixing it with the zein, it was even better than the commercial gold standards. Then we have the differentiation. So differentiation is the part when we have all of our cells, the maximum amount, we can't get any more. Now we need to force them to become muscle or fat. So there are different ways you can do this. You can either do it on the microcarriers that you started with, and then you'll have something that you can make the untextured products like the burgers. or you would transfer these cells to a new type of scaffold. And you could either do it on top of a scaffold for surface growth, or you could do it inside, which is called encapsulation. And this is what I do for my actual postdoc. So I'm using alginate, which comes from brown seaweed, and it's a polymer which, when you immerse it in a coagulation bath, it hardens. So, and it also swells a lot with water. So you can create these long fibers that are rammed full with cells. Then using special media, expansion media or differentiation media, so just as differential media for muscle or for fat, can force the cells to become muscle or fat. And we can see here some at the top, some multinucleated myotubes. And at the bottom, we can see some cells and the green dots are lipids. So lipids accumulate in fat cells. And with this, with the technique that I'm using, I can also make some aligned fibers. And aligned fibers could be great because alignment has been shown to help with muscle formation. So it might be possible for me to grow cells encapsulated to become fat, and at the same time grow cells on the surface inside the grooves between the fibers and create muscle. And that would be called co-culture. And I would be able to make some thick, realistic-like meat. But co-culture is pretty hard, and actually no one's really ever managed it properly, or at least at scale. It would be great because you can cut down the whole bioprocessing, because you only need one reactor now to do the muscle and the fat. And it would really enhance the taste and the flavor. It's just a much better process, and it's much cheaper, and it's potentially quicker as well. But the problem is that to differentiate cells into muscle and differentiate them into fat, you need different bioprocesses. You need different conditions. You need different molecules. You need different hormones. It's not that simple. So we have here UCL, Holly and Leo, who are PhD and postdocs, and they're going to try and solve this. So firstly, they're going to get some muscle progenitor cells and some fat mesenchymal stem cells, and they're going to grow them together in 2-dimension. They're going to try and find out exactly what makes it work. 2-dimension is the easiest type of culture, but they're going to try and find the right media, the right conditions. Let's hope it works. If it works, they're then going to move on to the 3D stuff. So they're going to do fibers like the stuff that I'm doing, and also microcarriers that will be encapsulated with muscle on one side and fat on the inside. And if that works, they're going to try and scale it up in spinner flasks and maybe even bioreactors. But this would be a very, very big achievement if they did manage. So let's talk a little bit about what's happening at UCL. So UCL has one of the largest cultivated meat research groups in the world. Definitely one of the largest in the UK, but there's not that many in the world, so still in the world. We have 11 active PhDs and postdocs. Most of us are working on bioprocess development and scale up for various tissues. We also have scaffolds, encapsulation, serum-free stuff, and computational models. If you came to UCL, you could also become part of the alternative protein community. We have a student society called the UCL Alternative Protein Project, which is a GFI managed but student-led society. And they have regular talks from academics, policy people, and industry leaders. They go see startups. It's quite exciting. It's a fun society. And then there's also, if you were to do a PhD or a postdoc here, you could join the Bite of Tomorrow network, which is part of the Bezos Center for Sustainable Protein. And that's really something that can advance your career to getting jobs in cultivated meat or advancing your academic research. And if you were to do a B-Eng or an M-Eng in biochemical engineering, you can also expect to have some stuff on cultivated meat and some assignments on it. One of the fun ones is the scenario week, where you'll be asked to manufacture imagine manufacturing your own project and how you will get there with the bioprocess. and you'll be helped with an expert in the field. So just to conclude, so cultivated meat is a direct response to the growing population and intensive farming by growing meat without animals. It builds on a century of funny predictions and advances in cell culture, biotechnology, food engineering, and a bunch of other sciences. In the last 15 years, that's when most of it happened. We had the first cultivated burger, the first steak, the first meat from an extinct animal, the first meat produced in space. We now have about 12 products that have passed some sort of regulation in the world, actually in only about 6 countries. But it might still be a while before we see it in supermarkets or in restaurants here in the UK. There is still an issue about scalability, and that can be achieved by producing serum-free or cost-effective media, getting better scaffolds to promote faster culture and differentiation, and also trying to get this co-culture right. So if you're interested in alternative protein or cultivated meat, you should start it at UCL. So thank you for listening, and I can now answer some questions.
Fantastic. I really enjoyed that, Ben. Thanks very much. What I'm going to do is we've got 3 questions that have come in already. I think one of them I'll never go answering that because that's more of a general thing about how many people are on programs. But there are 2 of the questions there for Ben. Please put your questions in the Q&A box. We're really looking forward to them. We've got a few to kick off with, but you can ask them anonymously if you want to. But this is your chance to quiz Ben. What I'm going to do is I'm going to read out the question from somebody. Somebody's asked how many master's students there are in UCL. Are they involved in research in any research groups? Okay, across UCL. There are several thousand masters, but that goes right across. UCL is a multidisciplinary university, so these will be people doing anthropology and languages and archaeology and science. So the MSc, there are many across very different parts of UCL. And the really interesting thing is that we sometimes get collaborations between something like biochemical engineering and dentistry. An example of really interesting collaborations between dentistry and art was somebody used images to be able to describe different kinds of pain. So because it's very difficult to find a way to describe pain, it's very difficult as a diagnostic tool. Whereas this artist was able to give like fire or sharpness, or you can actually use it as a diagnostic tool. And that's the advantage of being at UCL, is that you are in a true multidisciplinary environment. In our department, in biochemical engineering, we have 3 postgraduate programs. We have the MSc in biochemical engineering, the MSc in manufacturing, commercialization of stem cell and gene therapy, and then an MRes, which is MRes in synthetic biology, which is really it's a very research intensive program. So majority of the year you will be doing original research. Almost like the start off of a PhD, but you'll be doing original research in the laboratory. So it's a very focused program. The MSc Biochemical Engineering it's composed of modules that change depending whether you're coming in as a biochemical engineer, a chemical engineer, or a life scientist, and they really. So depending on what kind of background you've got, you can be brought up to that level. So it's a really interesting program, and the program tutor for that, Dr. Petra Hanger, is also really, really involved with cultivated meat, lab-grown meat, and has run a couple of conferences. But that's the background to that. I hope that answers your question about how many. Maybe I've given too much information, but that's a bit of information about master's students at UCL. Okay, Ben, over to you to answer whichever ones you want.
Yes, I'm just going to do the easy one first. So there was one about my PhD research and why I decided to use silkworm, silk fibroin as my biomaterial. So at the time of PhD, I did a literature review, found all the materials that could be used and which ones had been used in tissue engineering and had also potentially been used in food sciences. And silk fibroin is interesting because yes, it's from an animal, and technically it does have to be killed to get the silk. but it can be made recombinantly. So you could make it with bacteria if you really wanted to. It's too expensive to do now, but if sulfibrin was the way to do it in the future, you could mass produce it and make it really, really cheap and really, really pure. Sulfibrin is interesting as well because it's used in food as for packaging. It's technically regarded as safe for consumption. It's a protein, so you can digest it. And it's compatible with cells. Cells quite like it, despite not having any kind of motif that allows the cell attachment. It does. It's got other properties that help. It's quite hydrophobic. It's quite strong. It's got loads of other things that might help with cell attachment, but mainly that was it. It's something that's pretty cheap, pretty renewable, and that can be produced at enormous scales. Um, well, one question here is about which meat do I think will be the first to be sold commercially over a long term beyond the small scales so far in the industry? So, so far, most companies that have made it past the regulation have been making fibroblast meat. So, in this presentation, I've been focusing on adherent cells, which would be stem cells or muscle and fat progenitor cells. But you can also use fibroblasts. Fibroblasts grow like weed, and you can grow them without scaffolds. So you can grow them directly in suspension, but all you get at the end of it is more fibroblasts. They are differentiated a tiny weeny bit, but you're just getting more fibroblasts. Fibroblasts in vivo is connective tissue. They make connective tissue, and it's basically scar tissue. So if you consider scar tissue as meat, I think that will be the future of cultivated meat for the time being. Chicken is also much more popular. It might be that avian cells are easier to grow, but I don't know. I think it will be chicken, these types of foods that will be there for at first. But if co-culture worked and if it was done efficiently, you could get proper meat products very, very soon as well. So blood serum, there's a question here about where does it come from? Well, it comes from fetal calves. So you basically have to basically kill like late stage embryos from cows or really like fetuses. It's not great. It's actually pretty horrible. And that's where you get it. And that's it. So it would be great if we could move away from it. There's a question here about if I've tried cultivated meat myself and what was it like. So I have, but I had the Meatly product, which is the one that is sold in the UK, and which is technically dog food. As far as how it tastes a bit like a diet protein bar. It's mostly not meat. It's only 4% cultivated meat, 96% plant-based. And of those plants, it's mostly fruits like bananas, berries, and stuff like that. So it really is a bit like a protein bar, but not that tasty. But my dog really, really, really loved it. So I suppose the target audience likes it. So there's a question here about scaffold-free cultures. So yeah, if you go with fibroblasts, you can already do that, but there might also be another way around it. So you could use adherent cells and try to genetically modify them to be able to grow better in a suspension or produce more of its own extracellular matrix for other cells to form aggregates. But the problem with that is that you're making GMO, and that is a different problem with the regulation. In a future world where GMO is not a problem, I can see that being the solution.
So I'm just going to jump in and say, Ben, just because the questions are coming thick and fast, just tick them off once you've answered them. Otherwise, we might look like
how do I do that?
Great questions coming through. And also, I just want to say that if you go onto our YouTube channel, there are actually talks by Petra We also had Travis last year was talking about how viable is it to grow T-Rex legs and dodo nuggets. So we've some of this stuff coming up. We've gone a deep dive into it before. Anyway, Ben, back to you.
Yeah, let me just find some questions.
Sure. Actually, whilst Ben's looking through those, because they're coming through, we've got loads of questions to get through. We'll do our best to get through as many of them as we can. But on Monday, we've got a session with Matthew Banner, and he's going to be talking about AI in the pharma sector. So it's something that came up on Monday when we did our session talking about new vaccine manufacturing processes and synthetic biology. But a lot of people are asking about how we use AI in biopharma sector, and Matthew will be talking about that. He also works. He's doing he's done his PhD in collaboration with an industrial partner, so he's got a very interesting take on that. Cool.
Okay, so there's a question here about cultivated meat in space and how most companies are focusing on terrestrial or ocean animals. So there are reasons why growing in space could be interesting. So, for example, if you were to go for a mission on Mars or even much further, you would need a supply of protein. Now, you could do that with plants, but maybe astronauts want to have a steak every now and again. The problem is the conditions in space are very different. So you have to rely on microgravity or no gravity. And there's work to see how cells behave in those environments. It's been found that in microgravity, cells, I can't remember exactly, but cells seem to behave better. And so there might be some pros in doing it in space. But the other problem is that you also need a lot of energy to make cultivated meat, and that is going to be really, really limiting. So the resources, the inputs that you have to put to make cultivated meat might be a real issue now, and therefore completely unsustainable. But there might be some future avenue somewhere. So there was a question also about scalability for cultivated meat using electrospinning. So electrospinning is regarded by many people as not scalable, but that's just completely not true. When you're making it at lab scale, it's extremely slow. It's like you're pumping at 0.01 milliliter per hour, something like that. And so you're just going to get a small sheet this size over 2 hours, maybe. But you can do it with enormous industrial scale machines that can produce kilometers worth of these fibers, not kilometers of fibers, sorry, kilometers of mat in an hour as well. So you can actually mass produce it. Electrospinning is not the issue for scaffolds. Electrospinning is not necessarily the most common technique that where people are using for cultivated meat. A lot of people are looking at hydrogels, and so techniques to make hydrogel scaffolds could include bioprinting or wet spinning, which is a bit like what I'm doing for my postdoc. But you can make it through various ways. It's just some are a bit more scalable than others. So there's a question here about PhD opportunities at UCL. So the UCL research group is pretty big, and there's very often some research opportunities. In the last year, I think there have been 3 people joining either as a PhD or postdoc, but that's a lot for this type of lab. So there's a question here about cultivated meat in relation to ultra-processed foods and whether I consider this ultra-processed. Well, technically, we're making it ourselves, so it's de facto processed and even ultra-processed. But when people consider ultra-processed, they think of how food has been transformed so much with additives and MSG and all these other things that are generally considered unhealthy. Cultivated meat will just be meat. So there are studies that show that meat is not necessarily very healthy, but it'll be the same. And actually, by using different ingredients in your scaffold, in your biomaterials for your scaffold, you could actually make cultivated meat more healthy than real meat. So in my PhD, for example, I mixed the silk fibroin with psyllium husk, and psyllium husk has been shown to lower cholesterol for people who have high meat diets. It also helps with colorectal, not as a complete treatment, but it helps avoid stuff like colorectal cancer, which has been shown to be associated with red meat consumption. It also has some effect in diabetes. And basically, a lot of diseases related to meat are made less bad when you have psyllium husk. It helps with digestion and loads of other things. So you could make this type of red meat more healthy that way. You could potentially also do it with GMO. So that's answered. So one question here is about what could be the most disruptive outcome of this technology, and considering the barriers, what is one thing I'd say needs to happen to move it forwards? Well, I don't know if it's necessarily the most disruptive, but one of the barriers is farmers, people who do not want to lose their jobs over cultivated meat. It's probably not going to happen. Cultivated meat is not intended to replace traditional meat, but more to work alongside it to make meat just a bit more sustainable and feed a bit more people in a better way. But still, there's concerns that this is going to take people's jobs. There is a cultivated meat company, little startup. Actually, not sure it's a startup, but there's an organization which is actually founded by the daughter of Willem van Elen, the guy who predicted and tried to make cultivated meat happen. And she is working with farmers directly to try and integrate cultivated meat or alternative protein at the farm. So a farmer could have in one barn thousands of chickens, and in another barn he could be producing in a bioreactor some proteins that would help cultivated meat production, or even literally produce meat in that second barn. And so you might, if you could try and get farmers and politicians and various other groups on your side, it might bring things forward. For example, in Europe, in the EU, just a few weeks ago, they announced that meat produced by, that is just not meat, like alternative meats, can no longer be called meat. So that would be for everything plant-based, but it will also be for cultivated meat. So cultivated meat will have to change name, but it will also have to try and convince people that what you're growing, if you're growing cells in a body or cells in a lab, the end product is exactly the same thing. That's done. So the question here about whether co-culture exists in biopharma at scale. I'm not sure. I don't really know enough about co-culture, but it's very difficult to scale up. A lot of biopharma uses very expensive stuff because it's just going to sell it for a high cost. But when you're making foods, no one's going to buy your 250,000 euro burger or even your 50-pound steak. You might do that once, but you're never going to do that again. So things have to be so cheap, and making it in a cheap way at scale is really the challenge. So some people have claimed to have it done, but we haven't really seen any concrete evidence of it. So a question about what drove me to specialize in this area. So most people most of my colleagues, most academics or people in industry who do this, it's mostly because they care about the environment, they care about animals, they care about public health. For me, it's a little bit different. I just wanted to do something sci-fi related. I wanted to try and create things that had never been done before, or try and advance science in a completely new direction and make a contribution to that field. Obviously, I care about the environment, I care about animals, and I care about public health, but that was really secondary to the sci-fi stuff. So the question here is interesting. Whether I see the largest breakthroughs in the industry from academia or industry first. So cultivated meat is interesting because it's one of the only, well, it's one of the very few, at least, sciences that is really driven by industry. So before the first cultivated meat burger came, it was mostly academia, but now it's mostly industry. But the problem with industry is that it's pretty opaque. You try and, make your product. You're going very, very fast, skipping loads of steps. And it could be that you're doing exactly the same as someone else. And it could also be that you're going completely the wrong way. We just don't know. And whatever they discover, they're not going to really tell us. So academia is doing its thing in parallel, and it's discovering things that are completely pointless and also things that potentially could be used by industry in the future. So it's difficult to know which one will figure it out first. Probably the big breakthroughs that will be in the news will be from industry, but they will have probably used some techniques that were developed in academia first. Question here about what risks are associated with cultivated meat products. So there are risks to consider, and that's what the regulatory bodies are trying to assess when you try and move a product forwards. Firstly, there's all the ingredients from the media, the scaffold, anything that you put in, there's a chance that it's not really that edible, or it could be even toxic, or it could lead to problems downhill. For example, I used, originally in my PhD work, I was using methanol to cross-link my sulfibroin. So basically to make it stable in a water environment, I use methanol. But methanol, even in very, very small quantities, is extremely toxic. It would have gone away, and it would have 100% been completely evacuated from the scaffold and even from the meat later on. But just having it there to start with is not ideal. You could also assume that, well, we don't really know. No one's really eaten that much. There's very few people in the world who've eaten cultivated meat, and there's also not been any studies for long-term consumption of cultivated meat. So even though it's cellularly exactly the same, the cells might be producing slightly different proteins that have not yet been identified, and that could potentially also have some kind of toxic risk after large amounts of consumption. And also there's a risk that cells change over time. So this is not necessarily the case with immortalized cells, but with stem cells, and especially embryonic stem cells, they might over time change their aspect and their function. And if you don't end with what you started with, then your bioprocess has changed, and you can't guarantee you're getting batch variability, and you can't guarantee the safety long term either. So you would have to eliminate this. This is generally controlled in a work environment, but this could be something to consider.
So I'm just going to jump in because we're literally going to the last minute now. So I'm going to ask Ben to look through the questions that are left and pick the one that he most wants to answer. I think we can answer in the time left. But whilst he's looking through that, I just want to say a huge thank you to everyone who's come along for your questions. These have been brilliant, and we wish we could get through all of them, but we've done our best to answer as many as we possibly can. I want to say this has been the most engaged, most questions we've had in any of the sessions so far, which so well done everybody here. Ben, over to you.
Okay, I'm just going to answer two more. And also, if I didn't answer your question, feel free to connect on LinkedIn, and we can have a chat later on. So one question here is about the sterility environment and the fact that it doesn't have the bacteria from slaughterhouses. So this is interesting because we know that once a animal has been killed and the meat, the flesh has been removed, they immediately gets contaminated with bacteria in the air, and it would affect the well, not the maturation, but the post-death maturation of the tissue. We don't know how it affects it, but we know that it contributes to the texture of the food. Because cultivated meat is all done in sterile environments, you could assume that there is absolutely no contamination, and it'll be completely sterile and potentially sold in a completely sterile packaging. So the shelf life should be longer. Again, you could play around with the shelf life just by having different stuff in the scaffolds. Zane, for example, is antimicrobial, so it might help prevent contamination post-assembly as well. And there was another question here, which was about could it ever become sustainable to create meat using renewable energy? I think it can, but this is a future problem. You could potentially have solar panels across the entire surface of the moon connected to bioreactors, and just grow it there in an environment where you're recycling the water. You could potentially do this, but we're not there yet. You could also potentially plug it into a fusion nuclear reactor and just get unlimited energy that way. There would be ways you could do this. It's just there are no real practical ways nowadays. And that's a problem because when people do life cycle assessments of cultivated meats, they generally look at what the technology is like today or try and predict something in the near future. But cultivated meat is not for today. It's for the future or maybe distant future. So we need to try and think about how other supply systems of energy can help bring cultivated meat to market.
Fantastic.
Anyway, I'll stop here. Any other question, just feel free to send them over later.
Great, thank you very much. Somebody just asked about previous webinars. I'll send an email out following up when we've uploaded the videos to YouTube. You'll also be able to watch them back on Zoom if you can't watch YouTube where you are in the world. And I'll include a link to previous taster lectures as well, so you can see what we've done in previous years. We had someone talk from Hoxton Farms as well, talking about the challenges of manufacturing fats. And also I send a link so you can sign up to UCL, so you can stay in touch, so you can find out about future events. But finally, just thank you very much for coming along today. I hope you had a fantastic week. And thank you, Ben. Thank you so much. This has been a fantastic session. And yeah, I hope to see you all in UCL at some point in the future. It'd be great. But for now, thank you very much and goodbye.
Thank you very much. Goodbye.